Niacin's influence extends well beyond metabolism and energy production. It reaches into the realm of genetic control, where it plays a vital but often understated role in DNA repair and epigenetic regulation. At the molecular level, niacin contributes directly to the formation of NAD+, a coenzyme that fuels two key families of enzymes critical to genome stability: PARPs, poly-ADP-ribose polymerases, and sirtuins. These enzymes are deeply involved in maintaining DNA integrity, modifying chromatin structure, and regulating which genes are turned on or off, a process collectively referred to as epigenetic control. In this context, niacin isn't just a nutrient. It becomes a gatekeeper of genomic health, with implications for cancer prevention, aging, and cellular resilience. When DNA damage occurs, which it does constantly due to factors like UV radiation, toxins, metabolic stress, and normal replication errors, the body launches rapid repair responses to prevent mutations from accumulating. PARPs are among the first responders in this system. They detect breaks in DNA strands and catalyze the addition of ADP-ribose units, using NAD+ as a substrate, to signaling proteins, thereby recruiting repair machinery to the damaged site. Without sufficient NAD+, and therefore without adequate niacin, this repair mechanism slows or becomes dysfunctional. Over time, the inability to maintain DNA fidelity can lead to genomic instability, a well-known hallmark of cancer development and premature aging. Simultaneously, sirtuins, another class of NAD+ dependent enzymes, play a subtler but equally important role. These enzymes deacetylate histones and other proteins, thereby influencing chromatin structure and gene expression. In simple terms, sirtuins help determine how tightly DNA is wound and, as a result, whether certain genes are accessible for transcription. This has profound implications not just for day-to-day cellular function, but for how cells age, how they respond to stress, and whether they enter protective states like autophagy. When NAD+ levels are adequate, sirtuin activity helps silence harmful genes, activate repair pathways, and reduce inflammation. When NAD+ is depleted, often due to low niacin intake, chronic illness, or aging, these protective responses decline, making the genome more vulnerable to instability and age-related diseases. The connection between niacin, epigenetics, and aging is particularly compelling. Aging is not simply a matter of accumulating damage. It also involves shifts in gene expression, often in ways that increase vulnerability to disease. Sirtuins, activated by NAD+, have been shown to delay the onset of age-related pathologies in several organisms by stabilizing DNA, supporting mitochondrial function, and preserving the epigenetic landscape. In this sense, niacin acts as a nutritional regulator of how our genes behave over time. A sufficient supply of this vitamin helps maintain the fidelity of cellular programming and slows the drift toward dysfunction that defines aging at the molecular level. Despite this, niacin is rarely emphasized in epigenetic research or public discussions about gene health. Much of the focus in epigenetics falls on methylation, histone modification, or environmental toxins, while nutritional coenzymes like NAD+ are treated as peripheral. This is a significant oversight. Without the substrate that powers chromatin remodelers and DNA repair enzymes, no amount of external stimulation or genetic potential can compensate. The machinery of epigenetic control is only as effective as the biochemical fuel it receives, and niacin is a major source of that fuel. The implications for cancer prevention are equally serious. Many cancers originate from a failure in the DNA repair system, mutations that slip through due to overwhelmed or underpowered repair mechanisms. If niacin levels are low and NAD+ availability declines, the efficiency of both PARPs and sirtuins drops, making cells more likely to accumulate the kind of genetic mutations that initiate cancer. It's telling that some forms of chemotherapy now target PRP pathways, attempting to manipulate this very system, yet we overlook the dietary foundation of these enzymes. By ensuring consistent niacin intake, we could potentially support the body's innate ability to suppress early tumor development through constant, effective repair of genomic errors. This also raises questions about the narrow focus of modern nutritional science, which tends to prioritize overt deficiency symptoms or population-wide deficiency statistics, rather than considering the full spectrum of what optimal nutrition should mean. The absence of pellagra doesn't mean niacin levels are high enough to support robust DNA repair or epigenetic protection, especially in populations under chronic stress, with high toxic exposure, or facing metabolic disorders that place greater demand on NAD+ turnover. In these contexts, even a normal niacin intake might fall short of optimal function. Ultimately, niacin is far more than an energy vitamin. It's a molecular linchpin connecting nutrition with genetic integrity, cancer defense, and cellular longevity. Its work is largely invisible, happening at the level of DNA strands and chromatin coils, but its effects ripple outward through every tissue, every cell, and every decade of life. By elevating our understanding of niacin from a basic nutrient to a genomic guardian, we gain a deeper appreciation of how the quiet foundations of health shape our resilience over time.